Hello everyone, you are all welcome to lecture number 1231 and today's topic is very interesting topic, that is isolation and purification of flavonoid. The chemistry of flavonoid is very interesting and it has lot of medicinal importance. Lot of work is going on and some very important flavonoidal glycosides are playing very important role in health and disease. So first thing is, what is flavone? The word flavone, F-L-A-V-O-N-E, is a Latin word. Flavone is a Latin word and it means, the meaning of flavone is yellow color. So the word flavonoid is derived from Latin word, means yellow pigment, yellow color from flower, from any source of the plant material or even animal, because some of the butterfly, they have yellow pigment, and that pigment, it was found when test was conducted that it is flavone. And when I say flavone, means it is only benzo-pyrone nucleus, but if I say flavonoids, means structurally related compounds are there, and most of these flavonoids, they are found in combined state with sugar. Sugar may be glucose, may be ramenose, may be xylose, may be arabinose, and that's why they are known as flavonoidal glycosides. They are called as flavonoidal glycoside. Flavonoidal glycosides means the non-sugar part, that is called aglycol, and the sugar part is called glycol. So it is made up of aglycol plus glycol, and it is flavonoidal glycoside. I am not going much in detail about the glycoside because one of my lecture is there in journal, what are glycosides, different type of glycosides, O glycoside, C glycoside, N glycoside. So that you can go to my online lecture, you can get. Here mainly I am concerned with flavone nucleus. Now what is flavone nucleus? Flavone nucleus is the condensation of three nucleus. One is benzene, other one is pyrone, and the third one is phenyl. So your this flavone nucleus is the condensation, is made up of three units. One is benzene ring, aromatic ring. Second one is, that is your pyrone ring, and third one is phenyl ring. So this is your benzene, this is pyrone, and this is your phenyl. So when they are condensed together, you will get this nucleus. One, two, three, 4, 5, 6, 7 and 8. So this is your flavone nucleus. flavone nucleus, or the other name of this nucleus we may also call, because when we are counting the number, it starts from heteroatom oxygen 1, 2, 3, 4, 5, 6, 7, 8. So at 2 position there is phenyl ring. So the other name, IUPAC name of this flavone nucleus can be 2-phenyl benzo pyrone. So 2-phenyl benzopyrone nucleus is called flavone molecule, and sugars are attached, and they are very powerful antioxidants because they are phenolic in nature, and also they are very good anti-inflammatory compound. Also it can act to improve the fragility of your arteries and veins. That's why it helps with cardiovascular system. It can check and balance the growth of even cancerous tumor. Quercetin is well known nowadays, most costliest flavone sold in the market in Western countries. That quercetin is the most powerful anti-cancerous flavone nucleus, and also it has lot of other benefits for human health and disease. Now, once you know about the flavone nucleus, now how to test, how to know that in which plant material it is containing flavone or there is flavone molecule. So for that there is a specific test. That specific test means you take the alcoholic extract in the test tube, few milligram, dissolved in ethyl alcohol, add into it few pieces of magnesium chips, and side by test tube very gently add few drop of concentrated sulfuric acid. Immediately the bubble and reaction will start, and after few seconds the red color will start appearing. The appearance of red color confirms that flavone is there. Now it is confirmed that your alcoholic extract of any plant material is containing flavonoid molecules. After that you have to do the phenolic test. Phenolic test means it will give you the indication that there are some phenolic groups, means hydroxy is attached to aromatic ring, benzene ring. So for that you just take little extract dissolved in ethyl alcohol, and that alcoholic solution separate it, and in that just add 1 drop of alcoholic ferric chloride solution. Immediately it will become dark blue or dark black. Changing color into dark black or dark blue confirm that it contains phenolic flavonoidal compound. So after doing this test, then we are proceeding for isolation. How to isolate the flavonoid from the flower or from the root, stem, bark, leaf, any part. But the richest source of flavonoid mostly they are the flowers, and it can be there in any part of the plant. So you have to collect the plant material, dry it in shade, and then do the extraction with ethyl alcohol in the lab, and then evaporate all the ethyl alcohol. After evaporation you will get the concentrate of the extract, and that extract is there which is rich source of flavonoid, and now you are having the concentrate of flavonoid. After getting that, then it is isolated from plant. Now you have to proceed for purification of flavonoids. For the purification of flavonoids, you have to follow some chromatographic technique, and these most popular chromatographic techniques for the isolation of flavonoidal glycosides are chromatographic technique, that is thin layer chromatography, TLC, then column chromatography, and mainly these two, column and thin layer chromatography, they can help you to purify. And once they are purified with the help of chromatographic technique, then we have to proceed for the structural determination. And that pure compound will be subjected to spectral studies with the help of NMR, IR and UV. Then we will be in position to establish the structure. So I am going to write some of the structures of the flavonucleus. So here some of the very important flavonoidal structures are given, and my advice to all the students who are offering this course, that is flavonoidal chemistry, should practice these structures, and they are simple: 2-phenyl benzopyrone nucleus. Try to remember like this. The first nucleus like this. Start from here, 1, 2, 3, like that. So at 2 there is phenyl group. This is pyrone. This is benzene. So the first simplest nucleus of flavone is 2-phenyl benzopyrone. That is called flavone. There is difference of O and A. So here, this is your flavane. I mean, here it is not there. That's why it is called flavane. Number 2 is, that is flavanone. Not OA. Flavanone means here in pyrone ring there is no double bond. Then again in this nucleus, if at 3 position there is hydroxy group, then it will be called as flavanol. And if there is double bond in pyrone ring, then it will be called flavonol. So AO has big meaning. And number 5 is flavane, means where oxygen is not appearing. And then isoflavone means where the phenyl ring is shifted from 2 position to 3 position. One such position is there, then it is called isoflavone. And if this pyrone ring is open, then it is called chalcone. And number 8, where there are 4 phenyl group, 2 in phenyl and other 2 in benzene ring, it is called quercetin. And number 9 is camphor. Only difference is that here one hydroxy is less, and this quercetin, camphor, and the number 10 is epigenine. Is also having the similar structure like camphor. The only difference is that is the OH, and here and other OH is there. That's why it is called epigenine. And they are all medicinally very, very important, and they play as an antioxidant in our health and disease. So with this I will stop here. In the next lecture I am going to discuss about the sugar linkage in flavonoid and what are the specific compounds in the form of glycosides which are very important in our health and disease.